hour-long conversation with our esteemed panelists. Our aim is to explore the evolution of agriculture, tracing its trajectory from being considered an art and skill to its current status as a highly refined science. The panel discussion will delve into understanding how simple breeding methods like phenotypic-based artificial selection, hybridization, and mutation breeding evolved into advanced technologies such as molecular marker-assisted breeding, genetically modified transgenic crops, and gene editing platforms. Our focus will be on understanding the technological paradigm shift that transforms agriculture into an interdisciplinary field, supported by techniques like NGS, multiomics, big data, bioinformatics, machine learning, and tools like sensors, drones, satellites, and artificial intelligence. And how these technologies have helped to bring precision and smartness to plant breeding, and also reducing the time lag in the development of new varieties. With this introduction, I would like to start the discussion with Professor P.P. Gupta, sir. So, in the pre-industrial era, agriculture was practiced as manual labor, basic tools, and dependence on natural processes. It had limited efficiency and low productivity then, and this period is referred to as agriculture 1.0. Then, the advent of steam engines and tractors led to mechanization, and coupled with increased use of chemical fertilizers, it led to improved farming practices during agriculture phase 2.0 in the early 20th century. The visible agricultural growth took place in the third phase, the post-World War II, mid-20th century era, that witnessed the Green Revolution. It involved adoption of high-yielding crop varieties, extensive use of chemical inputs, irrigation advancements, and increased crop yields. Lately, in the 21st century, in agriculture phases 4.0 and 5.0, the precision and smart agriculture has emerged with the integration of digital technologies and all the other techniques like data analytics, sensors, GPS, which help in real-time decision making. Internet of Things, robotics, and automation has revolutionized farming practices. And now, the increased focus is on sustainability, resource efficiency, and customized farming approaches. So, with all this, agriculture has come a long way, and apparently, we have done a lot. So, you must have witnessed all these phases, in your lifetime. We have heard the stories like how in 1960s, the times of food scarcity and starvation, when India had to survive, they say from ship to mouth, as there was not enough food in the country, and the country had to wait, people had to wait until the next ship showed up. And now reaching to a level that we have surplus food, and we export food worth billions of dollars. I would like to ask you, sir, are we doing the right things in agriculture? Is all this sufficient to meet the UN goal of zero hunger by the year 2030 and to make Bharat Viksit by the year 2047? Or, in your opinion, are there some other facets also that we should focus at? Kindly guide us, sir.
Once again, I'm not very sure what I'm supposed to do, but one question which comes to my mind, there are several questions which come to my mind. One is that should there be so much emphasis on agriculture in teaching biology or what? The subject of botany, should it gradually become agriculture? I think should not. I've been a student of botany myself, and later on I entered into agriculture. But there are so many important areas in biology and botany other than agriculture. direction from the teachers of botany. Recently, new syllabus has been prescribed for BSc Biology. My publisher asked me to write few books in that area of new syllabus. And during the last 3 years, I have written 4 or 5 books according to that syllabus. I found that many people who are in the area of preparing this syllabi are themselves not competent really. And therefore, these syllabus which have been prepared according to the new education policy would be better if expert advice was taken. For example, the latest book which I have just finished writing, the title of the book is Cytogenetics, Nanotechnology and Digital Technology, something like that. There are about 15 chapters in this book which devote to nanotechnology, artificial intelligence and digital technology. I have myself had to read a lot in order to write these chapters, and I'm not really sure whether I have done it justice to this. And these areas, nanotechnology, artificial intelligence, digital technology, they are not only important for botany other than agriculture, but they are equally important. Major emphasis when you talk about nanotechnology, artificial intelligence and digital technology is really agriculture. So perhaps in this panel discussion we could think and discuss to what extent these new technologies, including nanotechnology, artificial intelligence and digital technology, they are going to play a major role in augmenting agriculture production and productivity in our country and at the global level. Because it seems that nanotechnology, artificial intelligence and digital technology, they have already playing a major role. Digital technology is very important because so much of data is accumulated. We have to make use of this data for not only generating knowledge, but also for utilizing it for a variety of purposes, including agricultural production, environmental climatic changes. So there are a number of these areas where the new technology involving artificial intelligence and computation, they are perhaps going to play a major role during the next one decade. But perhaps you could also discuss what are the other areas other than agriculture which botany students should really be taught. I think my colleagues here will perhaps have better wisdom to talk something else other than, but to me these are certain things which came to my mind and we can initiate with this.
Thank you so much, sir. Thank you for your insights. The Botany Honours curriculum in Delhi University, which is NEP Botany Honours curriculum, it does not have much of a component of plant breeding. We just float a paper, special paper of plant breeding. Whereas at times, students, they come forward and they ask us, like, please float that paper so that we have some know-how about, like, what happens in breeding also. So we have taken a note of your suggestions. Thank you so much, sir. I have another question, but I'll ask you after I finish this. I'll now move on to Professor A.K. Singh, sir, and then I will direct myself to Professor Penta. Professor A.K. Singh, sir, transitioning from conventional breeding to molecular breeding, you and other plant breeders in agricultural universities and institutes in India and across the world have been quite successful in selectively manipulating genes for developing crops with desirable traits such as increased yield, disease resistance, and adaptability to changing environmental conditions. You have already showcased this beautifully in your presentation. The precision of molecular tools has marked the onset of a new era in crop improvement that has not only expedited the breeding process but has also enhanced the resource efficiency and reduce the environmental footprint of agriculture also. Undoubtedly, molecular breeding offers practical solutions to challenges posed by a growing population, climate variability, and evolving pests, envisioning a future where crops are meticulously crafted to meet the dynamic demands of the world. I'll ask you two questions, sir. You have been working in rice breeding for past so many years, and you have developed so many improved varieties that are already commercially cultivated in farmers' food, which is very commendable. But most of these varieties have been developed in white indica rice. The diversity available in the form of land races of rice in northeast, Indo-Gangetic plains, and other parts of the world is mind-boggling. And these are varieties of rice like black rice, red rice, and purple rice that are highly nutritious, but they are very poor yielding. Are you also planning to genetically improve these important genomic resources? Also, sir, 70% of the millets and other marginal crops, they grow in India only. But they need attention and genetic improvement with respect to yield and taste enhancement. FAO of United Nations declared 2023 as the International Year of Millets to draw towards attention to this group of crops that got neglected after the Green Revolution. I would like to know from you, sir, what is ICAR's mandate on the genetic improvement of crops other than the staples like wheat, rice, and legumes, and especially the examples that I just cited?
Thank you very much. I will answer these two questions for sure, but if time permits, give me some time to answer the questions which you had raised to Professor P.K. Gupta. They are very relevant in terms of Indian agriculture. What is our expectation, where we are moving forward, and how. So coming to the first question, improvement of local rice varieties. As a matter of fact, we have about 10,000 land races which are locally named. They are collected, conserved in the National Gene Bank, and very recently we have taken them out, and through a project which was in fact strongly supported by Professor Pentel, we are doing detailed characterization of these 10,000 land races in terms of their value for biotic stress tolerance, abiotic stress tolerance, and quality. But in addition to that, there are a number of land races, about 200 rice land races, which are adored by local communities, and they are really liked for their quality. For example, if you talk of Eastern Uttar Pradesh, we have Kala Namak, the rice protected under geographical indication. Odisha has Kala Zira. West Bengal, if you go, you have Randhuni Pagal, Govindo Ho. If you go to Manipur, you have Tahao Paritan, that is Manipur black rice. We have very targeted program to improve the productivity, because major problem that all these land races suffer is that they are very low yielding, hardly 2 tons to 3 tons per hectare, and at that productivity they are not economically viable proposition for farmers to cultivate. So we have taken up a very intensive project, and I have developed 2 varieties of Kala Namak called Pusa Narendra Kala Namak 1 and Pusa Narendra Kala Namak 2. These are improved version of traditional Kala Namak, which is meant for 11 districts of Eastern Uttar Pradesh. And this is considered to be gift of Lord Buddha to the 11 districts, and this is protected under the geographical indication. The productivity is improved from 2 tons to almost 6 tons per hectare. Likewise, one of my PhD students has just submitted her thesis. She sequenced Manipur black rice, that is the cowparitan, which has got very high antioxidant potential. And there also we have been able to improve the dwarfing. We have incorporated dwarfing genes, and we have got dwarf version of Manipur black rice also. Govind Bhag also we are working. As far as International Year of Millet celebration is concerned, in fact, before United Nations General Assembly declared International Year of Millet 2023, India had celebrated in 2018 the National Year of Millet, and it was on the behest of Indian government that UN General Assembly adopted 2023 as International Year of Millet. We have a full-fledged institute called Indian Institute of Millet Research in Hyderabad, which works on sawa, kodo, kutki, kangni, jowar, bajra, all these millets. And improvement is primarily focused on disease resistance, drought tolerant they do have, and make it still better, and productivity improvement. So number of varieties have been released, and in fact we have got about 114 biofortified varieties. Many of these are coming from the millet group of crops with high iron, zinc, and other potential nutrients. Thank you.
Thank you so much, sir. It's very heartening to know that your genetic improvement programs are extending to all the land bases of rice. And I must call you again after the lecture on genetic improvement of land bases of rice. I'll try that soon, sir. Thank you so much. Moving on to Professor Painter, Deepak Painter, sir now. Sir, so far cotton is the only GM crop that is grown in India, which was developed by Monsanto. The first indigenously produced transgenic food crop, Dara mustard hybrid 11, commonly known as DMH11, has been developed by your team at the Center for Genetic Manipulation of Crop Plants under the University of Delhi. The research was funded by the National Dairy Development Board and Department of Biotechnology. We congratulate you, sir, you and your team for this exceptional achievement. DMH11 is a cross between the popular Indian mustard variety Varuna with an East European variety Early Lira 2. Since mustard is a self-pollinating crop, crossing was made possible by sterilizing one of the parental lines. In the DMH11 Varuna variety parental line, a Barnese gene was introduced to induce male sterility and to prevent the plant from self-pollinating naturally. Whereas in early Lira 2, a Barstar gene was introduced, which blocks the effect of Barnese, thus allowing seeds to be produced when farmers grow DMH11. A third gene, a herbicide-tolerant bar gene, was introduced in both the parental lines for herbicide resistance to confer tolerance in the plant to use of glufosinate. Glufosinate is a broad-spectrum herbicide similar to Monsanto's Roundup. Non-GM plants would die when sprayed with glufosinate. Sir, the development of DMH11 is significant as Government of India wants to build self-reliance in oilseed production to save the huge foreign exchange outflow of about $20 billion annually on edible oil imports. That's how perhaps India's Genetic Engineering Appraisal Committee, GEAC, approved the country's first ever environmental release of genetically modified food crop DMH11 mustard in October 2022. The environmental release is an important step towards future commercial release of this variety. Sir, I would like to know from you, if you could please throw light as to where do we stand now with respect to GM mustard variety DMH11 for its commercial release. Also, if you could just add, what is the future of increasing yield by GM crops in India on other crops also, other than mustard, so as to contribute to agricultural productivity for Bharat to become Viksit by 2047? Over to you, sir.
The government of the day finally decided to accept the GAC's advice. The GAC is now an appraisal committee. They have no powers to release anything. The release is only has to be done by the government. So GAC, after about 10 years of going through different biosafety tests, about at least 10 crore was spent on the biosafety test. Not many of them were needed to be done, but we went along because the country had no experience with these biosafety tests. And these were done in the best public sector laboratories. Now, we know that there is opposition to these genetically engineered plants. It started in Europe first, and the reason is very simple. These are not individual choice technologies. If you take a cell phone, this is an individual choice. Now, artificial intelligence, there's so many downturns. I was looking at your slide of what needs to be done. first you create the technology to make fake papers or fake diagrams or fake, and then you have to have something, knowledge to recognize those fakes. This is one of the challenges, the second challenge is there. But AI will run because it is individual choice. It's easier to spread it. The technology of agriculture, most of the urban people will say, even if there is one penny of harm, why should I take it? And who is really bothered about how much productivity you need, how much food India needs? Only Ukraine crisis got the high inflation in the world, food commodities all over the world. So we are walking on a knife edge in terms of agriculture. But it seems to be away from our conversation, away from, because the urban, like I would ask, what will GM mustard do to me as an urbanite? Will it reduce the cost of edible oil? I don't believe that the cost of edible oil are going to come down because farmers are not earning enough already. And somebody can say that it will come down, but it will stop them from skyrocketing if there is global shortage. So these are societal issues which only the critical establishment can take. The government has taken that decision, which I appreciate, because the government has also taken action on genome editing. Today, if you see, the technologies were developed, the genome editing, China was nowhere in genome editing. They have no paper in this area, period. It was all European and American contribution, which led to finally to the mechanism of, and for which Charpentier and Goodman got the Nobel Prize. Now, what are we doing with that technology? Fortunately, a critical decision was taken that this technology can be released, it's not going to harm anybody. SDN1, SDN2 type of genetic gene editing should be allowed to go through. When it comes to introducing new genes, which what Gupta mentioned as a big achievement, targeted gene introduction in the genome. That's a new gene and that will be followed by the genetically engineered material. But this is a very wise decision this government has done. But there are some unwise decisions which I cannot stand up and applaud. And one of them is reducing the Department of Biotechnology's budget by 26%. This is the industry in which we want biomanufacturing, we want therapeutics to be developed in that. You do all for startups. You do everything to encourage these young people to independently work and not only seek college or university jobs. That is well understood. And that's a forward-looking policy. But the question is, how with reduced budgets you can achieve great tests where your budgets are already low by international standards. So I am very clear, until and unless we make up our mind that we are going to use science and technology, we lost on electronics, for example. That was, the present government was not there at that time. So there is a general lack of appreciation of what science and technology can do for a country. No country has ever become prosperous without engaging with science and technology. And we will not be. We will remain a low-income, middle-income country. We may be fifth, because look at the number of people here, 1.49 already. So we may be in aggregates doing well, but individual levels we know what is happening. And we need to produce more, we need to be more productive in our crops and so on and so forth. So there are huge challenges which cannot be met without R&D. As far as mustard is concerned, I must tell the audience here that the hybrid DMH11, Dr. Pradhan, when he left IRI and ARS,
much job security in ARS. Dr. Singh was also with us at Terry. He found that Indian into East European mustard is heterotic. This is the first hybrid that we have developed. But there are so many other hybrids possible. And people refuse to, because DMH11, what is so magical about DMH11? We just put it in the trials to show that there is heterotic potential in the gene pool of, and today, when I was telling you China was nowhere in genome editing. They have more patents in genome editing today than the United States. So if you want to take up something in your life, take up with full zest and zeal. And as far as botany and agriculture are concerned, these are artificially created boundaries. Borlaug did an MSc in forestry. He was to develop himself as a forestry man. And suddenly, leave it. And suddenly he found that nobody wanted to go to Mexico. But he was willing to go to Mexico in some remote place and set up a center. So people require opportunities. People require some start to do things. And no matter how much theory you read, learning by doing is the best learning. So we have many challenges. I have no doubt about that. But in agriculture, universities, botany is not recognized. They will not be given a job. In botany, agriculture, people will not be given a job. What the person has read, what the person has studied, now rice has become a model system. Like Arabidopsis was a model system for dicots. Rice is both a crop and a model system. So a lot of people who are doing botany would like to work with rice to find some gene phenotype interaction, and like wonderful work has been done in Arabidopsis. We never did much in Arabidopsis except 1 or 2 labs in Hyderabad and in Indian Institute of Science. So we are not doing enough R&D. We are not doing enough science.
Thank you, sir, for raising this important aspect. We have been asked, to conduct such conferences and then prepare a white paper and to submit it to the VC. And we have been told that it might go to LD and then the PMO. So your recommendations and all the suggestions, will be definitely noting it down and we'll be passing it on with the hope that something comes up. Yeah, thank you so much, sir. Now I move to Professor Rana, sir. We are privileged to have with us Professor S.M. Rana, former professor and head Department of Botany, University of Delhi, and now emeritus professor at Department of Biotechnology, MIT University, Noida. Sir, you have been a celebrated plant cytogeneticist, molecular cytogeneticist, and molecular biologist of our country. As compared to animal cytogenetics, plant cytogenetics is considered to be difficult. And that's how perhaps we have just a handful of plant cytogeneticists in our country. One needs a lot of patience, an eye for minute details, and a deep understanding of chromosomal behavior during division and segregation. The reason why cytogenetical research is not considered glamorous, and it has had only a few papers. However, the information about meiotic behavior, pairing affinities, and homologous and/or homologous recombination is vital and foundational for any traditional and modern breeding and crop biotechnology programs. For the success of the gene transfers in any crop breeding program, it is imperative to maintain the fertility of the improved variety, which is directly determined by the meiotic behavior of the hybrid, pairing affinities, and the anaphase segregation, which gets ultimately reflected in the formation of balanced gametes and seed set. I remember the olden days in the lab between 1990 to 2000 when many a times Pendle sir would visit our lab in Delhi University in the evening and would discuss about Brassica cytogenetics with you for hours. When he was working on the development of mustard hybrids, and later Dr. Ranjana Kaul from our lab joined Dr. Pentel, in a collaborative cytogenetical research to work on mustard cytogenetics. Sir, you have made meaningful contributions in another facet of research which is extremely important for genetic improvement of crops, and that is delineating and cataloging the genetic resources, the gene pools in many crop species, and identifying the wild progenitors of important crops like Arachis hypogaea, Cephlar, Coffea arabica, and Vicia fava, to name a few, using cytogenetical, molecular cytogenetical, and molecular biological approaches. So we compliment you for making such meaningful contributions in plant sciences, which are reflected in your around 200 highly impacted research papers with more than 5,000 citations, 31 PhD, 17 MD students, and many national and international awards. So my question to you is, how has conventional cytogenetics contributed towards crop improvement goals? Further, how has the advent of molecular cytogenetics and flow cytogenetics in the recent past allowed to link molecular genetics and cytogenetics in more meaningful ways? In order to understand and manipulate plant genomes more explicitly, I request you to kindly throw some light on these aspects.
First of all, I am thankful to the organizing committee members who have invited me here for active participation in the conference. And I thank Professor Vijay also for some good words, because writing information for me. But anyway, thanks to you. Now the question you have asked is as far as I can recollect, because it's a topic where you have to cover both cellular cytogenetics or traditional cytogenetics and molecular genetics. But I will just try to remember and tell about what you have spoken, what you have asked me to say. Now, first of all, in any field of plant sciences, whether it is biochemistry, molecular biology, physiology, breeding, agriculture, or whatever, it is most important, I say emphatically, it is most important that you should know what is the cytogenetics of a particular species. Without that, you are going to do blunders. So, for any plant species, I am talking of endosperms, the knowledge about cytogenetics is a must, without doubt. Now, there are parameters of cytogenetic things which we have to understand. The first and foremost thing which comes to my mind is the determination of the gametic chromosome number, zygotic chromosome number, and most importantly, the basic number, which we often denoted 2n, n, and x. Unfortunately, this n, 2n, and x in many of the cytogenetic books are all rubbish. Sometimes they say 2n, and 4n, and 2x, or 2x is okay, but they are very incorrect written in many of the even genetics books. There are so many flaws. So anyway, we have to determine the zygotic number, gametic number, and the basic number. That's a must. And you will be amazed to find out that every plant species has a definite chromosome number. And every plant species has a definite chromosome share. That is all God-given or nature, evolution, how it has happened. And one species will have 6 chromosomes, another will have 1260 chromosomes. So the number, first of all, we have to determine what is the chromosome. Then second comes to my mind is ploidia. If the species is diploid or polyploid. Now if it is polyploid, then not only if it is polyploid, you have to have, it's a must, to know about the nature of polyploid. Whether it is autopolyploid, it is allopolyploid, it is auto-allopolyploid, or it is segmental allopolyploid. You have to know the nature of the polyploid, otherwise all your investigations will be of no value. Because the investigations which which you have to do, whatever work you are doing or whatever discipline you are taking, you have to know definitely what is the nature of polyply. And this nature of polyply is very important, especially in the angiosperms where we have 80% polyploids. Polyploids are secondary polyploids, but they are as much as to the extent of 80% in angiosperms. Then another point which comes to mind is karyotypic variation. This may be relevant in some cases or not relevant in some cases depending upon the species which you are dealing with. But in some cases it is of terrible importance. For example, I remember the BB genome, or I think Professor Gupta will correct me if I am wrong, the first BB genome or I think AO genome was identified in Bradby or Triticum estimum on the basis of karyotypic formula. I think there was one Indian Sarkar and Stevens, that paper. So that is still valid today. So what I am telling you, so as far as the karyotypic variation is concerned, it is dependent on what type of work you are doing and what is the morphology of chromosomes. In some cases the chromosomes are so small that you cannot make out any more karyotypes. For example, in brassica, you cannot do that thing. But in other cases, you can definitely see the chromosomes in a good shape and make it karyotypic. And then next, most important point also in the cytogenetics comes biosystematics. In biosystematics, what I mean to say is that biosystematics deals with the delimitation of natural plant biotic unions. Now this is very, very important, and for that biosystematics has to be done without doubt, or needs to be done, and it's a long and laborious kind of exercise, but it is a must. In biosystematics there are many ways to do it, but one of the most important ways is that you cross the two species, raise the F1 hybrids, and in F1 hybrids see the fertility or sterility of pollen, sterility and seed sterility in the progeny, and then what Vijay has told you, determine the chromosome association. Now that is very, very important. Many people, many of us in human beings cannot correctly determine the chromosome association. Very, very difficult and very, very cumbersome. But what I mean to say is, because of, there are viral formation or no viral formation, all are univalent in F1, so that will actually help us, and the fertility will help us to allot genomes for particular species. That is extremely important. That, for example, if I say there is a genome capital A and capital B, that means the two genomes are altogether different from each other. And if I will say capital A1, capital A1, capital A2, capital A2, or capital A3, that means there are genome these chromosome segments which are similar in some respects or dissimilar in some respects. So you have to know the genomic formula of all the important species. There is no alternative for that, and for that, I am not telling this is the only solution, but one of the major important parameters is biosystematics. Not only that, if you go to Gossypium table or Triticum table or Oryza table, you will find there are so many hundreds of species are there. Everywhere you will see on the right hand side or the left hand side, the genomic formula will be given. Because that gives an idea, because I am a plant physiologist, so I don't know what did you know. But I will know that this species is quite distinct from this species, which has AA and BB genome, and this species is not that different to this species because it has A1, A2, A2, and so on. So this is again very, very important thing. Not only this, from the breeding point of view, this is extremely important so that so far as you see, making better varieties of agriculture crops or crop varieties. So it gives us clue also what is the primary, secondary and tertiary. Having said that, then we can develop also anicolites. And these are of terrific value depending upon the material which you are using. And there are so many advantages of raising anicolites, but one of the most important things is that you can allot a particular character or trait to a particular chromosome because of having the anicolites. For example, Down syndrome, I will give you an example which everybody knows. You can see what are the phenotypic characters which a person is as a trisome for Down syndrome or Turner syndrome. You can find out what are the abnormalities and why it is so, because of this one chromosome is missing and another chromosome is a trisome. And so far as, for example, I will give you example, there are many trisomes developed or anicolites developed. One of the best example is wheat, Triticum aestivum, where the genomic formula is capital A, capital A, capital B, capital B, capital D, capital D, and the chromosome number is 42, and then 21 bivalents are formed. And in this case, Sears and many, father of wheat genetics, they have developed all possible 21 types of, believe me, monosomics, nullisomics, trisomics, and tetrasomics. These are all genetic stock. And they have these anicolites have given tremendous information which chromosome contains which trait, because that kind of change is found in the phenotype. So that means so far as the developing of anicolites is concerned is also very essential for the improvement of crop species or crop plants. Another comes to mind is the substitution lines and addition lines. Again, it is a very, very important step so far as the crop improvement is concerned. For example, I will just give you for the sake of students, I have capital A, capital A, capital B, capital B, capital D, capital D is Triticum aestivum, and capital R1, capital R1. That means R is a genome for Scalecidial, right? So I make addition line. So there may be 7 possible addition lines I can make. In one we have capital A, capital A, capital B, capital B, capital D, capital D, then R1, R2, R1, R2. In another addition you have R2, R2. Then in R3, R3, R4, R till R7 and 7. And all these, mind you, has phenotypic effect. Then they find why this, if R1, R1 is there, what is the morphology change? If it R2, R2 there, and these are all information which are gathered so far as the information is concerned, they are utilized for cropping. So having this R1, R2 and these addition and substitution lines are extremely important. Based on these all information, you can have lot of information so far as the crop improvement is concerned, and this information is taken care of by plant breeders. Not only that, I can give a lecture on 3 hours. There are so many examples based on this information. By chromosome manipulations, there are many varieties which have been developed in crop varieties. Based on these, we know what is what, and then we cross it, and then also break this variance in wheat by removing 5BL gene to incorporate homologous pairing, and then incorporate R1 genome. And what will happen there? So then we will find out what is happening there. So in chromosome manipulation and chromosome engineering, these are very, very important, the information which I have told you, and also the information for cytogenetics, for all. I am also, I don't know, in India we are, Professor Patel is criticizing some of the, you see, things here, but I am also adding the same thing as Professor Gupta also. Haploid breeding. We are nowhere in haploid breeding in India. haploid breeding to my, I don't know agriculture from, haploid breeding is nowhere. And where in haploid breeding, particularly there are so many advantages of production of haploid. I will not go into details of that, but one is hybrid sorting. One of the most important methodology. Hundreds of varieties have been produced, especially in rice. Where are we? We are nowhere in haploid breeding, and when we have given the technology to produce haploids, that has come from Delhi University Botany Department, and we are nowhere in haploid. Only thing is that we have got some problem in the production of haploids, especially in legumes. But there should be a kind of a, you say, mission mode kind of thing, pumping about crores and crores of rupees and develop the standardized technique for production of haploids in legumes of tremendous importance. And when there is very less genetic diversity among some of the crop species, haploid production is a must because each haploid is genetically one different from the other. Then another thing, induction of polyploidy. This induction of polyploidy has also given in many of the varieties, particularly of those where they are vegetative propagated and they are auto polyploidy. Now, having said this, these are all which are so far as the molecular side, cellular cytogenetic systems. I will do this point. Yes. So far as this molecular cytogenetic system, basically, for me, molecular cytogenetics is to see the chromosome from inside. And so far as cellular and this traditional genetics is concerned, cytogenetics is concerned, is looking the chromosomes from outside. Now, from the day the serious chromosome research was being done, every time, every cytogenetist used to bother about to linearly differentiate the chromosome. Many technologies have been well developed, but they failed. Then came what came banding pattern. Now people said that now okay, now we can linearly or longitudinally differentiate the chromosome. It was highly successful in animals, particularly in mammalians. It was not as successful in plants because in plants the most important banding. You pick up any human karyotype, photograph in the genetics, you will see the G banding pattern. Every chromosome has at least 20 G bands. You can map each and every chromosome depending upon G bands, and it is luxuriantly utilized for clinical cytogenetics in human beings. But unfortunately in plants, there is no G banding. You can't get G banding. And Professor Gupta was telling about chromatin and all kind of these technologies which have been developed. And on paper, to the students, we can say what is the difference between plant chromosome and animal chromosome. We say there is no difference. So far as the structure is concerned, it's exactly the same. But if it is exactly the same, what about G banding? You are not getting any G banding in plants. 2 or 3 papers were published, but there is a big question. And the point arises that G banding is not successful in plant, only C banding was successful to some extent, or N banding, other bands are not of that value. So far as the linear differentiation of chromosomes in plants is concerned, it was not that kind of a big leap forward so far as the banding pattern. Then the confluence of molecular biology, cellular cytogenetics, and of course image analysis gave a technology which was mind-blowing, and it was a breakthrough. That is what we call as FISH technology, fluorescent in situ hybridization. That was basically confluence of this conventional cytogenetics, information we have, and then the molecular biology, and then the image analysis. And this FISH, once it was developed, Immediately there are modifications in fish. You have now dish, you have fiber fish, you have prince fish, you have bath fish, and you have so many fish, fish techniques are there, and they are utilized in different forms, one way or the other way, in the system. Now, the time is very short. I will briefly mention. Actually, there are so many advantages on molecular side. Nucleus, what is happening, what is not happening, RNA processing not happening, whatever. So many, but I will not go into details of that. I will tell the points which are relevant here. Number one is, this is the first technique by which we can map the genetic, the genes or gene clusters, or we can say the DNA chromosome fragment. We can map the chromosome fragment, and we can take the repetitive sequence. There are 95% of the genome in high plants is nothing but repeated DNA sequences, highly and repetitive DNA sequences. We can map them very, very well now on the chromosome, and then see the difference between the chromosomes, where they are rich and where they are poor. So we can map these gene clusters on the chromosomes by fish. Then one of the most important things which happened is dish. In dish, you know, everybody knows that one of the most important things, when you want to have an improved variety or genetically improve a crop variety, you must know what are its what are its ancestors. So by many means we have done. So far we are also doing molecular biology, proteomics, metabolics, everything we are doing. Even then, in many of the species we don't know what are the ancestors. But Genomic Institute hybridization was taken into consideration, and dozens of papers were published only to recognize what are the wild progenitors of a particular tetraploid or hexaploid, and beautiful preparations were done. Now, for the sake of students, for example, you have an yellow hexaploid, 42 chromosomes. Genome is AA, BB, DD, RR. Now, when you will make a chromosome preparation, so far as histocarmine or Sildenstein is concerned, they will be all of the same color. But in dish, you can identify AA chromosomes very clearly from BB chromosome, genome chromosome, very clearly from DD chromosomes, and very clearly from RR. Beautiful preparation. You have no discrimination whatsoever. Beautiful preparation. When I am in off mood, I just see on the TV, on the laptop, see some dish preparation, I get elated. Such is the thing which I am seeing. Not only that, now the changes which are taking place, for example, there is, in case of Lycopersicon esculentum, they have found that there has been translocations during the evolution. From one genome to other genome, you can recognize it in dish straight away. Then you can have also the identification of addition lines, substitution lines, in which case you will find why is the additional chromosomes in the complement, why is the substitution line in the complement. So this gives all sorts of information us, and then so many numerous applications of molecular cytogen has come. Now there are so many papers which are published here in localization and detection of the introgression lines with the desired gene, carrying the desired gene. You may have a population of 200, 500. You can also identify which is the line where that particular introgression gene has been incorporated and what place has been incorporated to map it. And that mapping of introgression gene, this introgression gene, you are facilitating it by using microsatellites or mark libraries to find out why this is present. So then another, you can have also the translocation lines. You can detect translocation lines. There are so many advantages of translocation this is about molecular. Another technology came into being is flow cytogenetics. Now this is another fantastic kind of, this actually the technology which has been developed. And unfortunately, India is nowhere in both these, either molecular or flow cytogenetics. We are nowhere now in conventional cytogenetics. Forget about molecular and flow cytogenetics. Very, 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 very hurts me like anything. And what I mean to say is that flow cytogenetics, again, there are so many advantages. Only one advantage I will tell you here. For example, there is a plant species where there are 4 chromosomes, and in 4 chromosomes there are 2 homologous pairs of chromosomes. Now in flow sorter, this is the modification of flow cytometer, and there is a flow sorter there where you can isolate one chromosome from the other. So you have one chromosome, separately thousands of them in metaphase. The flow cytometer takes place in that flow in metaphase, chromosomes are flowed, and you have another chromosome, another homologous separately. So you have one chromosome and another chromosome separately one from the other. You can play the you want. You can have bulk DNA libraries of that chromosome. You can do whatever you isolate the DNA of that chromosome, whatever you want to do, and so many applications are given here. So this is in a nutshell what was the question posed to me. But in the end, I must say that it hurts me that there was a time when we were number 10 in cytogenetics in the world. People were recognizing that so far as this conventional cytogenetics was concerned. Now we are in gutter, we are nowhere in the dustbin, either for conventional cytogenetics or flow cytogenetics or molecular cytogenetics. The most important thing is that you submit a project because the person who are concerned there or the decision makers, they know zero about cytogenetics. They don't have any expertise in cytogenetics, and they are decision makers. You can do anything. And I talked to a person over a party. I told him there is a medicinal plant, Bacopa manora, Brahmi. It's universally medicinal plant. This Bacopa manora has 4 chromosome numbers: 12, 24, 26, and 30. Then there is another very, very important medicinal plant which is critically endangered, is Nordostatis jatamans. It has again 4 chromosome numbers. So, and I produced the literature, and when I saw that there is chromosome number so much of variation, I found there the papers published on active pollution. Tremendous variation in active pollution. And it could be related with the change, difference in the chromosome number. I asked one person, a very good scientist and very level. I talked to him, I said that I discussed this thing with him. I told him, why not submit a project, sir? Do you know what was his answer? He said, chromosome there is no way to get. This is his comment. So I just left that man there only and without bothering what nonsense he speaks. The same way, what I am telling you, I will tell you from 10 years onwards, I may be surviving or not surviving, the molecular cytogenetics is going in such a fast pace. Throughout world papers are published in scientific reports, science even now. And then the technology is developing so fast, you don't know what will come next in molecular cytogenetics and in flow cytogenetics. And I don't have the time. I will tell you that you can synthesize new species based on the information which you will get from molecular cytogenetics. Tremendous amount of interrogation is taking place. Tremendous amount. In some distant species, when you cross them, if they are able to cross, you will find that there is tremendous amount of translocation stages, interrogation stages. You get a chromosome which is completely different from the 2 parents. If you want, I can send you the photograph. And if you make it fertile, by any means, it's a new species. So you can develop a new species. So these are the these are the things. I think we can write to the person concerned, whatever I told you about the cytogenetic research in India. Not only that, the cytogenetics which I was teaching in Delhi University or Jodhpur anywhere, were a full paper of cytogenetics. Now you have reduced the cytogenetics into 1/4 of the paper. You can't do that. Cytogenetics should be one full paper. Forget about this. I have got all good words about this digital technology and all that things, but you should not forget about cytogenetics. You should teach the cytogenetics in full form, particularly in botany. Full paper I used to teach. And there was no time. We had to take extra class to teach cytogenetics. Now you have reduced it in a small part, just as you have done in taxonomy. Many other things you have done in embryology. Many other things you have done. This is big development. Thank you.
Thank you so much, sir. Thank you so much, sir, for giving us a detailed overview of how cytogenetical research is important and how basic subjects. I think all the experts are talking about that only, that we should maintain the scientific of botany as a subject. How biosystematics, how embryology, how cytogenetics, all these things are important. And how cytogenetics is so foundational to anything that we do about crop breeding. Thank you so much, sir. We are running a little short of time, otherwise I would have loved to discuss at length all these things. I'll pose 3 quick questions to 3 other experts we have, skipping their introduction because they have their sessions in the afternoon session. So we have Suman ma'am with us. Look, I cannot be formal with you. You are Suman ma'am. So, since we have discussed a lot of research-related questions here, Suman ma'am is an academician. She is head of our current department of botany in the University of Delhi. Apart from looking at the research and academic and administrative activities, she spearheads, the botany-related queries or the spectrum of botany activities in 30 colleges of Delhi University where botany is taught. So I would like to ask you a question as a teacher. Like, there is a call, ma'am, for Viksit Bharat at 2047. So what role do you think academicians and academic institutes like Hansraj College and Delhi University can play towards Bharat becoming Viksit by 2047? And also, since we are plant scientists, we are botanists, can we get some cues from the nature? Can we understand, like, nature also guides us? So do we get some cues from the nature and we can design, like, what all is supposed to be done in the years to come? Over to you.
Hello. Yeah. Thank you, Dr. Vijay. And first of all, I would like to acknowledge, I will be very brief, but I can't resist that, for making me a part of this wonderful event. And being in the company of such people is itself a great privilege for me. Let us begin with Professor Rana. They are the drawings of plant sciences. And Dr. A.P. Singh is presently the shining star in the field of genetics and agriculture research. And from my left side, they are the rising stars. I'm sure I can very confidently say I know Dr. Bhargava's work. So to be sitting in the middle of this kind of a galaxy of people is a great privilege to me. I really thank you so much for it. Coming to the teaching and particularly in the light of the new education policy, I've tried to make some notes. I would like to respond to Professor P.K. Gupta's very important observation that certain syllabi need drastic improvements. I totally agree with you. And we have the first review of the MAP syllabus, which has been implemented 2 years ago, after 3 years. things. So I'm sure we'll take note of all the things which have been mentioned here. Not me, my successors, I can see my younger colleagues sitting here, and I request them to, I urge them to please make notes of, I may not be there, I'm sure I will not be there at that time in the Delhi University, but you must make notes of all what has been, I think individually everybody has made some very important points on what should be included in the courses at the UG level and at the PG level, particularly keeping in view how things have changed. So one thing is, to keep a very careful balance between that we should not be losing out on the traditional things like taxonomy and cytogenetics and biosystematics. But at the same time, my requests are again things like AI and even because when I was talking about in situ and dish and fish. So every time there has been a newer technique, it has very significant inputs from people who are outside the biology, not even plants, not even botany. So these emerging techniques have come because people from physics or computer science or chemistry have actually given inputs to understand biology. So keeping this balance is very important when we are formulating syllabi, because these UG students and PG students are the future of the country and the world. So that is the major challenge, that not leaving the traditional subjects, but keeping at pace with the modern developments which are happening. And very brief, as the instructor said, learning from nature, it is endless. There are endless lessons nature has taught us. The first statement that comes to my mind when you say is that life is the most successful enterprise ever started. So if life is the most successful enterprise ever started, what are the features of life which has made it so successful? So the first thing again is collaboration, resilience. Whether we start from evolution of a eukaryotic cell, we started with a kind of a symbiosis between a bacteria and a very primitive cell. So collaboration, collaboration, collaboration. That is the key lesson which nature has taught us. That, and another thing I will say, the more diverse people disciplines collaborate, more innovative or better will be the product. So that is the biggest thing. And if you allow me to just take 2 minutes, please. I have worked for a very long time before I quit that area in a very different system, which was production of lac. Lac is a very important commercial product, very traditional product, everybody knows about it since Mahabharata days. It is a product of an interaction between plant and insect. So the collaboration has to be positive, not negative, because we know many pathogens and pests they attack the plants. But when there is a positive collaboration, lac, which was believed to be a very small and humble product, today is one of the most commercially important chemicals because it has more than 100 chemicals in it. And one of the very important chemical, allylitic acid, which is the precursor molecule for all the chemicals in the perfumery industry, which is one of the most expensive industries. That allylitic acid is obtained from that. And again, when we were studying this, we found that it is not only a plant and insect, there is a bacteria involved in it, which is actually giving all the amino acids to the insect, because phloem sap is not giving an essential amino acids to the insect. So the question was, where are the amino acids coming from? So there is a bacteria involved which is giving amino acids. There is a yeast involved which is giving some fatty acids. There is even a phage involved. So all across the kingdom, the interaction is leading to the production of lactic. So this is, I think, to me, is the biggest lesson from the nature, collaboration. And as diverse as possible. That's all I would
Thank you so much.
say.
Thank you so much for answering the question. I will quickly ask a question to Professor Pati. Professor Pati is Professor in Head Department of Biotechnology and Head Department of Agriculture at Guru Nanak Dev University, Amritsar. Sir, despite making such technological progress in agriculture, feeding 10 billion people by 2050 is a challenge even now. And most important now, we are talking about maintaining the sustainability of soil, water and environment. The way agriculture has been practiced over the years, this has had a significant bearing on the quality of air, water and land, which are the pillars of sustainability. Green revolution, it helped us a lot, but then it had a lot of negative consequences also. I won't get into the detail. And Punjab has borne the maximum brunt of green revolution. Not only the economic aspects like the quality of soil, water, the water table has gone down, so much of irrigation, so much of pesticides, so much of fertilizers, catching up with the next cropping cycle, burning the stable plant. This has so many problems. So I would like to, since you come from Amritsar, I would like to know from you, what are our strategies to handle such problems which have arisen because of green revolution, especially in Punjab and adjoining areas?
Thank you. I'm blessed to be here amongst such stalwarts. I mean, we have grown as a researcher, reading the papers and books of Professor Gupta, Professor Pendle, and Dr. Rana. Extremely blessed to be here. Coming to this question, I think we have a lot of undergraduate students here, and my answer to this particularly, I'll put it in a very simple way, just to understand what we need to do in this kind of situation. The only mantra, what Madam asked me, the only mantra here is far-sightedness and innovation. In the morning we heard Dr. Tendal, Dr. Singh mentioning about the green revolution, and the crux of this is the far-sightedness. Whether it is Professor Barla, whether it is our Professor Swaminathan, this is far-sightedness. And thinking about this, bringing to this germplasm to the country and the whole era of green revolution. The other aspect which is important is innovation. I'll give you a small example. A contented society will not innovate. A contented society is not going to take risk. Risk is taken when you are pushed to the corner. And all innovation has taken place when you are pushed to the corner. You might have heard about the story of Pokhran 2, when India did a nuclear blast and the whole world was against us. I mean, India was in a very critical juncture because India was pursuing its space mission and all, and we wanted the technology from outside. And nobody was there to help us. Even our closest friend like Russia was not willing to help us. And I still remember that in the Times of India, that time we were doing our PhD, in the Times of India, the front page, angry Indian did it. So that was the kind of resolute that we should have. We can do everything, but we need to understand how our system can help us. So I am a teacher, I am a professor, I am teaching in a university. The sad part is, gradually, the critical thinking among students is gradually diminishing. What we are imparting is a monologue. We are just, how do I spell DNA? What is genetic engineering? We are taking classes, but the critical thinking is not there. If I ask a student, what are the major 5 challenges in Indian agriculture, or for that matter, for Punjab, they will unable to answer. That is the state of affairs. So, when I say contented society, when Madam asked me the problem in Punjab, there are challenges. In fact, 80% of this is, we have marginal land farmers in our country, less than 2 maybe hectares of land. So, farming is not any more profitable for this. They have to adopt new strategy, and they have to go for crop diversification, and they have to use maybe corporate might come here going to do anything. Crop diversification, they are not going to do anything because they are contented, they are getting MSP from wheat, they are getting MSP from rice, and these are the two crops the farmers are growing in Punjab. And all issues, whether it is water, whether all environmental issues, that's because they are growing all these crops, these two crops there. If you tell them that you grow low-yield apple, we have demonstrated them you can grow, they say, no, we are getting 40,000 rupees in 1 acre to the land. So, whole system had to change. I mean, the triple helix has to be developed, the society, farmers and the researcher, that should be a kind of what Madam said just before me, a strong collaboration and critical thinking, brainstorming, where all stakeholders has to be in one platform and discuss about the problem, and probably we can find a solution to this problem.
Thank you for your remarks. Okay, the last question to Dr. Telapilla Bhardwaj sir, who is the Principal Scientist in Division of Genetics at IRL. Dr. Bhardwaj sir, you have been credited with the release of first drought-tolerant introgression variety in chickpea that is developed through marker-assisted backcross breeding. This variety has made news and has been acclaimed by Bill Gates as well. How do you feel that the genomic revolution will pave the way for future crop improvement in India and elsewhere?
Thank you. Because of the right to pay respect to Dr. Gupta and Dr. Seniors, who were my teachers here. I would like to put it in two phases, the genomic breeding technology 1 and the genomic breeding technology 2. Whereas in the genomic breeding technology 1, where we concentrated primarily on genetic resources, the characterization, evaluation, which now has to move into fixed approach, focused, intensive germplasm selection approaches. The second was the development of genomic resources and breeding methodologies. The advances in breeding methodologies of self-pollinated crops. The third was genomic assisted breeding, where already India has taken a lead. Now going to the GAP 2.0, we have published this in Trends in Plant Biotechnology. I would like to say that apart from genomic selection, where we are looking at increasing per se the breeding value, because the relative genetic gains over the last decade has relatively decreased. What Professor Pantel Saab has shown us over from 1960 or 70, excellent gains, but over the last 1 decade. So we need to totally increase the breeding values and how we are going to bring about by per se using converting the breeder's equation into genomic estimated breeding value. Genome introgression, excellently put both NSR, where cytogenetics and the basic cytogenetics will play a role. New species or deselecting the old species already. Russian lab is working a big way into this. The third, what I say is the genome editing and the transgenic technologies. But apart from 3 G's of this, but E, gene ecologies, where artificial intelligence, where bioinformatics, handling of the large amount of data. We don't have good, what we call as a bioinformatics, people who can lead to handling of even biological data, leave aside agricultural data, and it is traveling by amount of gigabytes a day. So these are the things that are going to rule, and at the same time when we start looking at Sigma 6P, that includes quality of agriculture, quality of environment, which includes water, soil, as well as air, quality of education, that is going to play a major role, where the teachers now become mentors and we become mentees, so that there is an interactive interaction to understand actually with a focused dedication, quality of social life, that is very, very important. So these are some of the things that will be paving way for educational as well as agricultural development. Thank you.
Thank you so much. So we'll conclude this panel discussion, and I would just like to highlight that Government of India has defined the Agri Agenda for Ambedkar, which directs to raise farm productivity of grain crops and release that land to cultivate more fruits and vegetables and other high-value crops to raise farmers' income. This needs to be done without harming the soil and using less water, and in ways that are resilient to climate change. A lot of emphasis has been laid on regenerative agricultural practices now for sustainable and resilient farming system, ensuring holistic ecosystem health, soil regeneration, carbon sequestration, and biodiversity conservation. It is crucial to strike a balance between crop improvement for desired traits and preservation of the genetic biodiversity. Undoubtedly, agriculture has come a long way and there is much more on the horizon. By finding new ways to apply agricultural technology to one of the most pressing needs, we will find answers to tomorrow's questions and make advancements in the future of agriculture. With these words, I profoundly thank our illustrious panelists for sharing their knowledge with us with such enriching panel discussion. With this, we come to an end of technical session 1, and I hand over the dais to Dr. Pooja for further instructions. Thank you, everyone.
Thank you so much, everyone, for this brainstorming panel discussion. Your insights and contributions made the session truly enlightening and engaging for everyone.